Hola, buenas tardes. Buenas tardes a todos nuevamente. Estamos muy contentos este segundo día porque vamos a tener otra charla muy interesante. Yo soy Juana Barca y aquí está la compañera Valeria.
Hola a todos y todas otra vez. Ojalá que estén disfrutando estas sesiones de charlas. Hoy tenemos a un investigador de la Universidad de Salford y el Museo de Manchester. La charla va a ser en inglés y vamos a aprender un poquito sobre la herramienta de ADN ambiental en la conservación de mamíferos, aves y en específico anfibios. Más que todo anfibios, pero esta herramienta se puede usar para muchas cosas, como van a ver. Y sí, con mucho honor y mucho placer le damos la bienvenida a Matthew O'Donnell.
Bueno, gracias. Matthew es investigador del Manchester Museum y él va a acompañarnos en esta tarde. Como dice Vale, la presentación es en inglés. Así que cualquier duda, ¿verdad? Que tengan, al final podemos aclarar si tienen alguna duda. Matt, hello.
Hello.
Good afternoon. How are you?
Good afternoon. Yeah, I'm very well. I'm very excited to be here today and to discuss some of our research, some of our work that's currently happening at this very moment in time. So it's coming to you live and direct, which is always exciting.
Well, I know you very well, but most of the people here maybe not. Can you explain a little about your work? And I don't know, you going to present right now?
Yeah, I can start with the first slide and then kind of introduce myself. Okay. That probably makes more sense.
Okay.
Okay, so
okay,
perfect. Yeah. Excellent. Thank you. Thank you. So yeah, hello everyone. My name is Matthew O'Donnell, and I am the curator of herpetology at Manchester Museum, and I'm also a PhD student at the University of Salford, studying environmental DNA metabarcoding. And today I want to talk about the trials and tribulations of eDNA sampling in Costa Rica. And this is the research that myself and Juan have been conducting alongside Valeria and many others across Costa Rica over the last few years. It's still very much a live subject, so I don't have as many results to share with you today as I would like, but we are in the middle of a very extensive surveying effort at this very moment in time. So we're going to be discussing some results to date, but also the exciting possibilities of this technique and its application here in Costa Rica. I'll also be happy to answer any questions at the end, should any of you be interested in learning more about the technique and also my other research as well. So yeah, my research is supported by Manchester Museum, which is part of the University of Manchester in the UK, where I work. And it's also supported by the University of Salford, where I'm based in my PhD as well. So a big thank you to them and their support. And if you would like to follow me on Twitter or X, I suppose, as it's called now, that's my handle down there, Matthew O'Donnell, where I don't update as frequently as I should, but I'm sure there'll be lots of information when the final results of this work is conducted. Okay, so I thought I'd start off with a small introduction into frogs in danger and why in particular we are focusing the research that I'm conducting on this group. So we now know that there are approximately 8,500 or 8,679 species of amphibians, to be exact, at this moment in time. That number is always increasing, in no part thanks to Juan and Vale's recent description. And this is a very diverse group, but it also is a very vulnerable group. We know that at least 40% of known described amphibians are at risk of extinction. And this figure is constantly changing as we develop new techniques to survey and monitor amphibians. But what we do know is that there are a toxic cocktail, as I like to call it, of threats that are facing amphibians and that are causing these risks of extinction. Now, this can be things as that we're all familiar with, such as the climate crisis. It can be deforestation. It can be habitat loss, habitat degradation, a whole host of threats. But it also, in one large area, is the invasive fungal pathogen, chytridiomycosis, the disease that is wiping out a lot of amphibians. And we know from the Sheila et al. Paper that came out a few years ago that over 500 species of amphibians have been directly impacted by this disease, with approximately 90 extinctions. So as a panzootic, it represents the greatest loss attributed to a disease, to a single disease. So if we were to put that into perspective with mammals, we would be pretty terrified, I think, to be honest, to be frank, if we were seeing losses at this level in a group such as mammals. So the fact that it's in amphibians means that it doesn't necessarily garner the same attention that it would, say, if this was mammals or birds, for example, in groups that tend to be a bit more enigmatic and loved by the general public. However, we should care, and there are many reasons why we should care. Least of all, amphibians are often described as the canary in the coal mine, which is, for those of you who aren't aware, back in the olden times when they used to do a lot of mining, especially around the UK for coal, and they would take a canary, a small delicate bird, down into the mines with them. And this basically was an early warning sign for the miners that if a small bird such as a canary was singing away, they knew that the air quality was safe for the miners while they were down in these deep caverns. Now, if the bird stopped singing, that might mean that there was a poisonous gases being released by the miners that would be undetectable to the miners initially, until it got to a level where it was really dangerous. But the bird, being more susceptible, more vulnerable, would be kind of affected first. And that basically is what we describe as an early warning signal. It's pretty brutal in its application, but it goes to show that some species are more vulnerable to environmental factors than others, and amphibians are one of those. The fact that they have for the most part, a dual life of an aquatic larval stage and a terrestrial adult stage means they are dually at risk to environmental factors like pollution, like habitat loss and climate change. But then you introduce these invasive diseases, and it really does end up being the last straw, which unfortunately many of these species cannot tolerate. So we are losing a lot of amphibians, and those that aren't losing, that aren't even immediate at risk of extinction, we're still seeing severe declines. So yeah, there is a lot of work to be done with this group to try and protect them for the future. Okay, so why Manchester and why Salford? So I'm very conscious of the fact that I'm sat here talking to you as a European, as a native of Manchester in the United Kingdom. And why am I talking about Costa Rican amphibians, and why am I talking about extinctions in these parts of the world? Well, through my work, I work in a collection of live reptiles and amphibians in the vivarium at Manchester Museum. And I've been there now for coming up on 10 years. And a lot of that collection is focused on the tropics, primarily the neotropics, so a lot of Costa Rican species. We have a population of Atelopus varius, Aglatinus anni, Aglatinus lima, amongst others that are threatened species. And we've been part of conservation projects now in this part of the world for several years. So through that work, we've developed a good understanding of the husbandry and the work that goes into maintaining these animals in collections. But also, we've been very much inspired into helping to protect them in the wild. And a lot of our work is inspiring our visitors to support conservation projects and fundraising to help protect these animals in the wild. So there is a history of working with amphibians from this part of the world through my work at the museum. And then at the University of Salford, where I'm conducting my PhD, we have a specialist eDNA facility. So for those of you who aren't aware, the image here with the forensic looking laboratory outfit is my supervisor, Dr. Nayara Sales, in the eDNA laboratory at Salford. Now, this is a very specialist facility, which is a completely clean room. So to establish this, it costs a lot of money, but it also needs a lot of kit. So you can see on the wall behind her here, there are UV tubes, and these are emitting UVC. So they're very dangerous to people, and we only use those while the room is empty, but it completely sterilizes it and destroys all the DNA that is left in the room. So through having this specialist facility, we've been able to do some really exciting environmental DNA projects across the UK, across Europe, and even in places such as Brazil, Vietnam, and elsewhere. Because we have this facility and because it's at a university, we're also able to do these projects at a scale and at a price point which is very much accessible to small projects such as this one that we're conducting in Costa Rica. And that's really important because there are facilities that offer environmental DNA services, but they tend to be quite expensive. So for small scale amphibian conservation projects, they might be a bit too expensive to approach. So through having these facilities and having these collections that I've been working with, I've been able to develop this technique using the collections in the museum to a standard now where we're able to use it in the field and be confident with the results that we gather, that we are seeing what we're seeing, and that we can say with certainty which species are present and absent within the environment. Okay, so another justification then. So I was very fortunate to be invited to be a part of the IUCN Red List assessment that was conducted in Costa Rica in 2019. And this is an image of the report that came from the end of that meeting. This was a really inspiring sort of workshop that was conducted studying the risks and the threats that are facing Costa Rica's amphibians. And through doing that, we were able to categorize all of the species that were described up to that point. So at that stage, there was 215 species. I now know that's increased. However, we were able to ascertain that approximately nearly 40% of Costa Rica's amphibians were either threatened with extinction to some degree or data deficient. And one of the takeaways that I came away and many of our colleagues did from the meeting was how many species that we just did not really have a true picture of their sort of current condition in the wild. And there's many reasons for that. Some of these species are very cryptic. They're very difficult to observe in the field. They're also difficult to find and to survey for, especially. And also they can be found in some of the most sort of remote and inaccessible areas of Costa Rica. So if we are to conserve some of these amphibians and to help protect them, we need data on their distributions, we need data on their occurrence, and we need that to be fed to the right people who are operating conservation projects here in Costa Rica, because that is really basically the building blocks for any conservation project. So how have we done this traditionally? So for the sake of a really good case study, I thought I'd talk about the Holdridge toad. So this is a species that I'm sure many of you are very familiar with. It was declared extinct and then rediscovered by our very own Juana Barca in, I think it was 2008. And this is a species that I think kind of epitomizes the struggles that we can have with traditional monitoring. So they are a fossorial toad that spends the majority of their life kind of underground, so in moss or under the the leaf litter, and for the most part actually in the soil. They're mute, so you're very difficult to carry out sort of acoustic surveys for them, and they tend to be very cryptic even when you do find them. You can see this beautiful specimen here and how well it kind of camouflages in with the environment. And yet this is a critically endangered species that's very close to the brink in terms of its prospects in the future. But what we do need to know is the full extent of its current range. We know where where Juan has found them, and also where they historically occurred. But we don't know if there is any other areas throughout that range where they might still be clinging on. We know of this one particular site, but again, this is very limited. So in order to maximize the chances of this species, it's important that we get a fuller idea. So traditional survey methods work fantastically well for many species. However, for species such as the Holdridge toad, there are limitations. And I think this is where things like eDNA, these tools, can become really valuable to conservationists. So what is eDNA? I've talked quite a lot about this, and I probably should have introduced this a little bit earlier in the slides, but the concept is effectively quite new. When we talk about eDNA, we're talking about DNA that is within the environment. So this is genetic material that has been shed from organisms into the environment. So that can be soil, water, even air, remarkably, which is containing tiny fragments of genetic material. And initially, when people were using the eDNA approach, they were looking at species-specific approaches. So using qPCR with primers that were designed to target very particular species. One of the first applications of this was detecting invasive American bullfrogs. And however, we've moved quite a long way from that stage now, where we're into really exciting territory of metabarcoding and beyond. But the original application was purely to find whether a species was present in a particular habitat or absent. And this is used a lot by ecologists in the UK to study some of our threatened species, some of our rare species of newts. And it still has its value in this day as well, you know, where you're working in particular regions where the diversity maybe isn't as large as it is in Costa Rica. So in the UK, species-specific approaches still have their value. Now, this beautifully illustrated example of what we're looking at here is particularly all different life stages of amphibians shed genetic material into the water. So the adults that maybe aren't within the system, but maybe live in the trees or the vegetation around the water bodies, they will shed their material just as much as the spawning or the tadpoles or the animals that live in around it. It isn't just from their skin or their mucus, it can be from their fecal material and even from dead specimens. So if an animal has died in the pool, stream, river, pond, or in the substrate around it, that material is often washed into the system where we collect the water. And the water is the primary environment which we focus our study on. So for other studies, they're looking at air or they're looking at soil, but in the tropics, especially here in Costa Rica, we're focused on eDNA from water samples. So I'll talk a little bit more about the methodology a little bit later, but just to give you an idea, it's basically collecting bottles of water, about 500 ml per replicate, from each system that you identify as a potential area of interest. So what is eDNA metabarcoding? So as opposed to the single species approach, we're now talking about a multi-species approach. Now, this is a really exciting application where we're looking beyond some, one target species into a particular group. So we could be looking at all amphibians to scales where we're looking at all vertebrates. So the benefits of this are, we're talking about non-invasive techniques. So we don't actually have to handle or enter the environment to a great degree. We can do this from a very easy sort of collection of water from a stream or pond. It can detect cryptic species, so species that maybe evade traditional surveying techniques. It's cost effective, so it's efficient in that manner. We're not going and spending days, weeks, months surveying in remote areas of national parks. We can go in, we can collect samples, and we can leave. And actually, we find that through standardized methodologies, we can get really accurate levels of representation from the data. So although it might seem quite remote that you would actually detect all the species that occur in a habitat just from water samples from one day, we actually find that it can be almost as comparable as sort of weeks and weeks of sampling in traditional methods. So it is a much quicker and cost-efficient method of surveying. This makes it especially relevant to amphibians due to their ecology. So the fact that amphibians might be quite elusive, many species live high up in the canopy that can be difficult, or underground. The fact that we can detect them from water is very promising. The specialist facilities, so I've talked a little bit about this development at Salford. So the fact that we have these clean rooms and these facilities where we can do all of the preparation is a massive advantage, because this again reduces the costs. And that is one of the biggest barriers to entry when we discuss eDNA surveying is the cost, because it can be quite expensive. However, it's scalable. So if you're doing 10 samples, it will cost you exactly the same as it will if you do 100 or 200 samples. So the fact that the costs go down the more you sample means that it favors larger surveying efforts, such as the one that we're conducting at the moment in Costa Rica. And then, yeah, we'll talk a little bit more about this as we progress. So our current sampling methodology consists of 4 national parks and 1 forest reserve within the area de Conservation Central in Costa Rica. We've sampled from an elevational range of 60 meters to 3,000 meters above sea level. So it's a massive variation in altitude and species that we're hoping to detect. We've done a total of 65 sample sites, including 15 field blanks. So we take a field blank on every single day that we sample. And that's a total of approximately 340 samples. So if you break that down, at each sample site, we collect approximately 5 replicates. So that's 500 mils per replicate from each site. And that is an awful lot of water to filter. It might not seem like it, but the reality over several days is very sore hands and very tired researchers. So we filter all of these samples as quickly as possible. And the reason for that is the environmental DNA degrades within the bottles. So even though we've captured it, it doesn't last forever. So the idea behind quickly filtering is that we capture that genetic material that is in the water bottle, and then we preserve it. So to do that, we use a preservation buffer that will store the DNA and prevent any further degradation of the sample. So we do that. We then label each individual sample. We sort of tag that. So we have the location for each sample. So it represents its geographic and topographic sort of sampling location. And this is really important data, because what we don't want to do is get back to Salford, do all of this work, and then not know exactly where these animals were occurring. So we have really important and vital information that is stored with every single sample. So we have all of this saved so that we can refer back to each individual sample and even the replica, and we can distinguish how many species were detected within each. And this is really important. And it's based off of science that has been done over the last sort of 5 or 6 years, looking at the amount of water that needs to be filtered, the amount of replicates that need to be taken in order to get a real representation of the species that live within the habitat. So it might seem quite random why we take 500 mils or why we take 5 replicates, but it is based off of evidence that's been conducted in these parts of the world, looking at similar sort of species groups as well. However, the reality is this is still a new technique. So although we have really good ideas of how to maximize and best sample these regions and maximize our results, we're still learning as this tool develops. But the important thing for me is that it has a conservation application. I didn't want to focus my research too much on the theoretical. I wanted to take the method and robustly test it in the field in Costa Rica, where we can put it through its paces by testing it, all different elevations, all different habitat types, all different systems from rivers, streams, ponds, puddles, lagoons, and really ascertain just how powerful this technique can be when applied as robustly and widely as we have done. So we're really excited about this. As I say, we're conducting this sampling right now over the last couple of weeks. So we're still quite a while away from getting the full picture of what we've detected. But it's been a real privilege to be able to visit some of these beautiful locations. We've been given really amazing permission as part of our permits to access some really remote areas of the national parks. And this has hopefully given us some amazing data, which will be of a benefit, not just to amphibian conservationists, but potentially to others as well. So a little snapshot of results to date. So this is based off of some pilot studies we carried out last year in Braulio Carrillo National Park. What you might immediately notice here is the very small number of amphibians that we detected. Now, I can explain this. We had some issues with our original amphibian primers. So this is a primer designed to amplify amphibians, where it actually was amplifying an awful lot of bacteria as well from the environment. So these systems that these amphibians live in, by dint of having amphibians and other animals living in them, also have a lot of bacteria. So we were having some issues with that we're trying to refine. So instead of waiting and not testing, we really wanted to see just exactly what was in there, maybe not to the same resolution, but just to get a picture of what species we were detecting. And to do this, we used a more generic primer, a 12S primer that was designed to amplify all vertebrates. And through that, we've detected a total of 850 MOTUs. So these are molecular operational taxonomic units, which don't always mean distinct species, but they're groups of organisms that share these kind of molecular characteristics. So it'll be certain sequences within the target gene. And when we actually go through and break that down into full species identifications, we had 3 amphibians, 19 birds, and 27 mammal species. So you can see that the vertebrate primer does heavily amplify mammals and birds over amphibians. Now, this is the case. We were expecting to get that sort of bias towards mammals and birds. Amphibians are especially quite difficult to amplify, and that's why you usually tend to use more specific primers that just target them and kind of ignore all the other vertebrates in the environment. But don't fear, we do have some really exciting primers that have just been produced that we're going to be trialing with this other sample set. So we're much more hopeful about the results moving forward with regards to amphibians. The other thing to bear in mind is the areas where we were doing this sampling. We do know that the amphibian diversity in that region isn't the highest as it is in, say, the lowlands, where you would expect to see a lot more amphibians. So although that we were detecting amphibians, and this is a really positive result, it's just a small snapshot of what we're trying to achieve moving forward. And then trials and tribulations. So I know I talked a little bit about tribulations in the title, and I thought this would be a good opportunity to talk about it. And I think this is true for a lot of researchers and a lot of people who are conducting research, especially when you first start out in your research careers. Don't get disheartened when things don't go your way. That's my advice, because we've had lots of really positive and incredible successes and moments throughout the years, but we've also had some setbacks. And these pictures illustrate a couple of those, where we had a freezer malfunction, where some of our stored samples were stored and they got defrosted, which is really problematic. So now we use this buffer, which preserves them even without a freezer. So we now have kind of circumnavigated that need for cold chain storage, which is a really positive thing. But that came off the back of this freezer malfunction. And when the freezer malfunctioned, it got put outside, and then raccoons got into the freezer and ripped open all of our samples. So completely contaminated all of our hard work that we'd conducted over the previous month. So that was one of our big trials and tribulations. And the other one was some of our equipment that we got lost in transit. So these kind of things are often big setbacks, and they can kind of build up and make you feel like, oh, is it meant to be? Is the research just never going to happen? And my advice is to always persevere, because you never know what's around the corner. And based off of this bad luck that we had, we've made some really important innovations in our sampling and our preservation. I did some studies at the University of Manchester, and we studied a variety of different techniques to preserve our samples. And that's where we isolated this particular buffer as being one of the superior options for sample preservation and preserving the DNA of the target species for long durations without cold chain storage. So even out of the gloom and the upset from these kind of issues, you can make some really positive decisions, and positive research can come out of that. So yeah, that's my 2 pennies worth on trials and tribulations. For the most part, it's been a really exciting journey, and something I'm delighted to be a part of, and I'm hoping we're going to get some really exciting results moving forward. And yeah, why is this exciting? So just to be clear, because I think this is often misconstrued by eDNA scientists, and I wouldn't necessarily view myself as an eDNA scientist. I view myself as a herpetologist, a science communicator who's also a molecular ecologist. I wear many different hats, but I do see that this is only one aspect that we require to tackle the larger conservation challenges that face amphibians. It isn't going to solve every problem. It's not a miracle. However, it does have great potential. But I do think that the biggest potential for this technique is when it's used in concert with other techniques, such as bioacoustics, such as traditional monitoring or remote cameras. These techniques are really important and have value, and it is just as much as the environmental DNA. However, when you use them together, I think you get a better picture of exactly what is going on in these areas, because there are species that I don't believe we will detect through eDNA, because they maybe never go anywhere near these water bodies. So a lot of the salamanders or direct developing amphibians, where we won't necessarily get any traces of them within the water systems. But if you use bioacoustics, cameras, or other applications of eDNA where you sample soil or moss or air, then you might actually get a full picture of the biodiversity in these sites. It works. So that is an important aspect. We know that environmental DNA metabarcoding works, and it works in the tropics, and it works in mega biodiverse regions such as Costa Rica. It's scalable, so we can roll this out into large areas. 65 sample sites is an awful lot, and that's been conducted over 2 weeks. Imagine if you were working for months, you could feasibly do massive areas of Costa Rica, massive areas of any country, and get some really amazing data. What we do need is money, and we do need resources and time. All these things are really important, but the fact is we can scale this. And one of the biggest ways we can do that is by ingratiating citizen science into the project. So citizen science, as I'm sure many of you are aware, is how we get the public involved in science research and projects. And this has been conducted by many of my colleagues at the University of Salford, where we've trained up volunteers and members of the public in eDNA collection techniques so that they can go out and sample large areas of the country, filter all of those samples, and then send them to our lab where we can analyze them. And I think that's one of the big hopes for this technique, is because even though we're showing the value of it and we can go out, there is only so many of us researchers out there, and there is so much, more to be done. The challenges facing amphibians and other groups are kind of monumental in scale. But I think by using technology, embracing machine learning and some of these other things, we can build tools that actually are kind of fit to the scale of the challenge. So yeah, citizen science, I think, is a really exciting component of this and could really broaden the scope of these projects moving forward. The fact that it's non-invasive is a massive bonus. So obviously a lot of these areas we've been visiting are incredibly pristine and really important habitats for many species. And historically, traditional sampling could be quite invasive. You might have to catch species to identify them. You might need to take samples. Stuff like that is still very necessary. But through using eDNA, we can maybe minimize the amount of invasive work that needs to be conducted, because we can use museum specimens to build a bigger picture of the biodiversity through the genetics that they hold. Big data, once you have these data sets, they can be used not only by herpetologists or conservationists, they can be used by all sorts of different people, because all the data that is produced from these is open access. So it means that it can be analyzed by researchers investigating all different types of issues, whether it's conservation, threats, chytrid, these kind of things. Having access to these large data sets is really, really valuable. And we know that, we have to be collaborative, we have to work together, and hopefully by producing this work, we'll be able to benefit many different species above and beyond the amphibians that we're initially targeting. And then there are future sort of applications. So the possible population level data and abundance data is something that is particularly exciting for amphibians. So we know that we can detect presence and absence, but essentially there are papers that are looking into, and researchers that are looking into, the possibility of getting data on a population level for species. So we could have a look at the diversity within the genetics to ascertain the sort of species, the scale of the issues facing that species, whether the genetic diversity is restricted, which means maybe there's inbreeding, and also abundance data, so we could get an idea of exactly how big these populations are. So I think this is really exciting as an application for the future of eDNA. It's not something that we're particularly at that level of being confident to roll it out, but it's something that's in the pipeline that's really, really exciting. So a big thank you. This project is the definition of a team effort, and it would not be possible without the tireless support of many of my friends and colleagues, particularly my supervisory team of Dr. Alan McDevitt and Dr. Nayara Salas, and the project partners, Juana Barker and Valeria Aspinall at Halakohaila Conservation, and a special thank you as well to Alex, Harold, Claudia, and Emmanuel from UNA, who've been amazing assistants and helping us with our collections over the last few weeks. I hope that gives you a little bit of an idea of the work that we've been conducting and some of the applications of this technique. And I'm happy to answer any questions about the scale of the challenges that we face. So yeah, thank you very much.
Thank you, Matthew. Thanks a lot for this talk. Very interesting for all. So let me check. We have a couple of questions. First, we like to share a small video about the techniques in the field. So I can share you in the field for people to see it. It's the first, and you have also a video for the technique. Right? Yep.
Yeah.
Okay, let me show. I wanna put off the presentation And I want to share this small video. Okay. Okay, this is Matthew in the field in the Braulio Carrillo National Park, and a very wonderful river. And we can explain very short the kind of stream or rivers that we are looking for is any kind of rivers or any kind of water system. Okay, so you can explain.
Yeah, so we're basically when we identify a sample site, we're looking at collecting a representative sample from that stream. So this is quite a large river here. So what we're essentially trying to do is maximize our chances of collecting genetic material from all the species that are used in this system. So that means getting water samples from maybe the fast flowing water, some of the slower pools at the side, and also minimizing any contamination that we might introduce into the system. So what we do is we use clean plastic 500 ml bottles, and when we collect the water, we use gloves to minimize any contamination that's introduced into the bottle. And we also then sterilize the bottle after we've collected it to make sure that any genetic material on the exterior is also removed. And we also have to be very careful not to walk within the system. So if we're walking through the forest and then we step and walk through the water or the stream, we're potentially contaminating that stream or pool or river with genetic material off of our boots. So not only is it really important to be biosecure in terms of prevention of spreading diseases through these habitats, so we have to be very mindful of sterilizing our equipment, but we also have to be very mindful of contaminating our samples to an extent where we don't have the same confidence that what we are detecting is actually present in that system, because there are chances that you could be introducing something from your boots or your clothing or your hands into that system that actually doesn't occur there. And if we want to use this data for conservation, we need to be very confident.
Okay, and the last video is here, filtering process.
Yes, so as you can see here, we're filtering every single one of our samples through a 0.45 micron Stereovex enclosed filter. So these are secure filters that will filter all the genetic material out of the water sample. And it's quite labor intensive, but we've made great efforts to reduce the amount of plastic that we do need. And the plastic that we do use, we do make a big effort to make sure it's recycled. But as you can see, the water is collected then into buckets, which we then sterilize before disposing to make sure that we don't spread any chytrid, for example, between sites. So all of that water, each 500 ml, is put through one Stereovex filter, and then we end up with 5 filters for each sample site. Those filters are then preserved with the buffer capped and sealed, labeled, and then individually bagged before being sent back to Salford and the labs.
Okay, excellent. So we have also a couple of questions. Let me keep off this small video. Thanks again for the video. So we have a couple of questions. Alex Valverde, good afternoon, Matthew. I hope you are more rested now and the sampling is done. I have a couple of questions. Are you planning and applying other methodologies like air sampling here in Costa Rica? And also, it was easier to get the permits for SINAC to DNA compared to more invasive process. And how did people react to eDNA? I mean, maybe he's mean about SINAC and DNA permits.
Sorry, Juan, you broke up a little bit there towards the end, so I didn't get the second part of the question, but I did get the first part, so I'll answer that to begin with, and then maybe the connection will improve. So we are interested in testing some of these other techniques. So my supervisor, Dr. Nayara Salas, is an eDNA expert, more so than I am, and she is very interested in the application of air DNA, so sampling from filters that we can put within sort of vacuums that are just drawing the DNA out of the air through a filter that you then preserve and extract in a similar method to what we would do with an aquatic sample. But we're also interested in the application of drones. So this is one area that I think is really interesting, where you can attach air filters to drones and then fly them through canopies. And this is a really interesting application that could potentially detect some of the canopy-dwelling amphibians that are traditionally very difficult to monitor. So things like Ecnomia hyla and Cruzia hyla that potentially are very difficult to see and might not visit the same water bodies that some of the other amphibians that we're targeting do. So we are very interested in those approaches for sure, although the current permits that we have don't extend to that. So it would maybe be a future project to study those.
And also the other question was about the permits. It was easier or it's complicated for DNA compared to normal otter procedure?
So I can only speak from my personal experience. We did have issues because the original permits that we were given were due over the pandemic. So we had these amazing permits to do our sampling, and they expired before we managed to do any sampling. So that was one of the issues that we encountered. We also had an issue then when we were reapplying for the permits. Obviously, although it seems that what we're doing is non-invasive and we're just collecting aquatic samples, we now know that the CONAHEBIO still viewed the genetic material that we gather. It's viewed the exact same way that tissue would be. So that kind of process means that it can be quite a lengthy application, but the permits that we were able to get in the end are brilliant. They're really extensive, fantastic permits. We were very grateful to be given. So it was a challenge at first, but pandemics and other things can't be helped, and we've really sort of made the most of the permits that we've now been given.
Yes. Now I think it's easier to get the permits for that kind. We have more experience about that.
Exactly. Yeah. And to be fair, the technique is so new that when you're initially talking about environmental DNA, not many people actually are familiar with the technique. So yeah, we now have that relationship where we can explain what we're doing really well.
Exactly. So we have a couple of questions more. Álvaro Figueroa, how can we get training as citizen science? I have access to the Isla del Coco or Cocos Island, which is pretty pristine. And also related to this question, there is another from Alex also. Is viable to have training people for sampling remote location for further minimize the cost of the methods? This question more about citizen science using this technique is able to share or to people can learn about the technique.
Yeah, so from my experience, the best way to do it is to run workshops. So as you're familiar, Juan, because we've done a lot of this together, there are a few little parts of the technique that are very important to minimizing contamination, but also to maximize the results that we get at the end. So the best way to do this is through workshops. So we do a lot of training, potentially, well, especially in the UK, where we've run a variety of workshops for many years with volunteer groups. And then once you get the volunteers trained to a certain level, they can train other volunteers. So it kind of grows exponentially. But I think the best way is to be taught by someone very familiar with the technique. It's a difficult thing to do via videos, for example, because I think a lot of it is subjective and it requires a bit of knowledge of the systems and also of the animals that occur within them. But I think it's something that we can do very easily through workshops, I think is essentially the best way. And in terms of getting people involved, I'm hoping that hopefully next year we'll have some really exciting results to share, but then we will also be hopefully running some workshops in Costa Rica that we can probably extend out to, you know, maybe 100 people or more. So if we do that in the next 12 months, I'll be sure to make sure that the invitations are spread far and wide. So anyone who is interested in the application can come and learn about it.
Okay, Matt. No, excellent. So we have time for two more questions, and we have two more questions. Alejandro Valverde asks, thank you, very interesting. I have a question. What do you attribute the low finding of DNA for amphibians obtained? Did you expect higher number when start the project?
So that is a really good question, and it was something that we were very conscious of, to be honest, when we began, because the areas where we were sampling we were in the highlands of Braulio Carrillo National Park, which traditionally did have a large amount of amphibian diversity, but subsequently it's you know, a lot of that diversity has vanished. So we are using the technique to cow in the background. We are using that technique to look for rare and endangered species, but with that comes the real possibility that you won't detect anything, because there might not be anything within those streams, within those rivers that you're sampling. What we did detect, however, are really strong signals for some of the more common species that you encounter in those areas. So Ismahyla pseudopuma was detected in lots and lots of our samples and corroborated our visual encounter surveys that we conducted at the same time. So we were positively identifying the amphibians we found in the system. It's just that the systems we were sampling were potentially devoid of many of the amphibians that once were within them. Whereas this surveying effort that we've done now is far, far more extensive. We've sampled so many more rivers, streams, and ponds, pools, you know, from low elevations where there's much more diversity to the higher elevations where there maybe isn't as much. But because we've sampled a more extensive area, I'm very hopeful that we will get a much stronger amphibian representation in the results. And then finally, just to clarify, one of the big things that I think is also hampered the identification of amphibians within our samples is the fact that a lot of the species in these high elevation areas do not have reference sequences within GenBank. So the fact that we can't assign, or the bioinformatics can't assign them to a species level, does not necessarily mean they are absent within the data. If you looked back at that slide where I said we had the motus, there was quite a lot of motus for amphibians, and it could be that within those are species that have not been sequenced. So that could be a lot of the animals that we were targeting. And that is one of the issues with eDNA metabarcoding, is that it is incredibly reliant on reference databases for the sequences that we're targeting, whether that be 16S or 12S. We need more of those sequences to be available. And that will probably involve a lot of work with historical museum specimens to sequence those and get those reference sequences available through GenBank, so that when we are doing the sampling in these areas, we will know exactly what is present and not, you know, present or absent.
Exactly, it's more genetic work for us. Exactly. And the last question is from Donald. Donald Varela is asking about how long the DNA stay in any species stay in water. Compare rivers to lagoon. How long, more or less?
So another brilliant question, and something that we have done a lot of studies in, we as in eDNA sort of researchers. So the baseline that we tend to go off is that we can still detect species up to around about 28 days post removal from a system. So that's based on aquariums and studies where tadpoles and fish have been put into an aquarium, removed, and then they've tested the water every single day until that species is no longer detected. And for the most part, it was almost a month that the DNA persisted within the environment. So that's really good to know that we have at least a month to detect the animals. However, that's based on a very particular set of environmental parameters. So that's temperature, pH, UV exposure, and all of these factors we now know degrade DNA at an accelerated rate. So the higher the temperature, the lower the pH, the more the UV exposure, the less time that DNA is going to persist within the sample. So we know at least that if we do detect the animal, that there is a real high possibility that it's been there very, very recently. And that's really important, that we're not detecting animals that maybe died there 20 years ago but have been preserved in the sediment, because we're not testing the sediment, we're testing the water, which has a much quicker rate of DNA degradation. So it's really important that we know this, but exactly how long the DNA will persist is determined by the environment. So that is, you know, specific to each pond, stream, or pool, because the pH, the temperature is slightly different in everyone.
Yep. No, excellent. Thank you very much for your answer. So we have 1 or 2 minutes to say goodbye, to say thanks for all people. First of all, thanks to you, Matt, for this talk. Amazing for all the work. We are hoping to finding very exciting things in the future. And this technique will be used for so many projects or other projects in the future. And well, a todos, muchas gracias por haberse conectado y por las preguntas que estuvieron muy interesantes también. Thank you, Matthew. You can say something more about.
Yeah, thank you very much, and I hope it was interesting. Please stay tuned to find out the results of what we find. I think it will be really interesting, not only for the herpetologists amongst you, but for the conservationists in general. I think a lot of people will see the value of this approach to surveying, and we will be sure to share the results far and wide as soon as we get them. So yeah, thank you very much for coming along and listening to our research.
Nos esperamos ver el próximo año con los resultados, o en 2 años en una próxima charla en Spanish. The next talk, the next 2 years maybe.
Yeah, exactly.
Thank you. Gracias de nuevo. Entonces estamos despidiendo esta presentación, agradeciéndoles de nuevo por todos los que estuvieron presentes y recordarles finalmente que estamos en la semana de celebración de Tlalocoila Celeste. Mañana tendremos otra charla también por el canal de Naturalista CR, más enfocado en lo que hablábamos un poco de ciencia ciudadana y cómo estuvo relacionado esto con la aparición o el hallazgo de la ranita Tlalocoila celeste. Recuerden también entrar a las redes sociales donde tenemos tanto los concursos como las rifas y bueno, todo es para apoyar este proyecto de conservación, Tlalo Conservación, que está dentro de la Fundación Costa Rica Wildlife. Pues nada, muchísimas gracias, que tengan muy buenas noches y bueno, los esperamos. Gracias, Matt, y hasta luego a todos. Pura bien.